Thanks, Peter, and thanks everybody for coming to this session. And thanks for the sponsors of this session. This is hopefully going to be a fun session. I am, as I mentioned, as Peter mentioned, I'm Dave Engelthaler with the Translational Genomics Research Institute. I have the enviable or unenviable job of kicking this off. I think it's enviable because there's a lot of great talks, and you're going to hear about how whole genome sequencing and next generation sequencing is really advancing in the field of food protection. My job is to understand who you are and to try to provide a bit of a primer, a bit of an update on whole genome and next generation sequencing. And I don't know if who's all in this group, if you can wax eloquent about metagenomics and bioinformatics, or if some of you are, don't even want to spell DNA. My guess is you all are somewhere in between. And so hopefully this won't be too redundant or too detailed. I think there's a lot of fascinating areas in how we can use whole genome sequencing. I personally am a public health guy. I worked for about 15 years in public health service. I was the state epidemiologist in Arizona. I worked for the CDC and have been in that field for a while. But I've moved over into genomics, which was actually fairly different for me. But I wanted to make genomics have real impact on public health. So rather than this title, which was really given to me, this is my title, which is How I Stopped Worrying and Learned to Love Sequencing, a shout out to Kubrick, but an understanding, and hopefully that you guys can understand, if you're reluctant to step into sequencing, you can see where it's going and hopefully see that it's becoming more and more accessible and inevitable. It's not, and there, Peter was telling me, what hype? And I agree, there's no hype. This is the real deal, and this is where we're moving, so it's better that we all understand where this is going. So before we start and jump into sequencing, I do want to talk just a minute or two on some basic pathogen genomic analysis concepts, just to make sure everyone's on the same page, or especially if I use acronyms that aren't totally familiar to everyone. So one of the concepts is genomic diversity, and we need to really understand where genomic diversity comes from in order to better understand what we find and try to describe how it got there. So bacterial pathogens, like a lot of microbial pathogens, divide, and essentially they carry their genomic material down to their descendants, and they carry it on forward. And then occasionally mutations will occur as they divide, and they don't get corrected by the correction machinery within the bacterial cell, and that mutation quite frequently will stay with the organism and get passed down to its lineage. So what we see is really at the very end here on the right-hand side, that's all we ever see. We see these variety of microbes out there with diversity in their genomes. What we don't see is everything in between. How did this get here? And that's what we're very interested in looking at and trying to understand. And by deciphering the genomes, we can start to understand the relationships between each other and understand what caused that diversity. And one of those mutational events causing diversity are SNPs. And hopefully you all know what SNPs are, single nucleotide polymorphisms that are, in essence, just the change of one of those DNA letters, one of those A's, G's, T's, and C's into another. Again, there was a mistake in the error correction possibly, and allowed then for this mutation, this SNP to carry on down. Sometimes these SNPs will be what we call non-synonymous and will end up changing the organism, change the gene that'll make a different protein, it'll change the color or the flavor or make it antibiotic resistant, somehow change the organism overall. And many times it doesn't do it. It doesn't make any kind of a change, what we would call a synonymous SNP. Overall, though, these are really important for us to decipher, to track inside the genome and then try to decipher what that means to relationship between organisms. And I'll show you many examples of this. I know you guys can't see the details on here, but you can envision that these are strings of letters, and these are sequencing reads going across, and we're comparing sample X, Y, and Z to a reference, and the reference genome is the one that's in the middle. And what we're looking for then is, when do you see a difference between the sample and the reference? And we can see in sample Y here right in the middle that this is a different letter than what is normally seen in the reference. That's what we call a SNP. And we can look across then the millions of letters and find these SNPs to help us understand, what are the mutational differences. And it's not just looking for SNPs, it's the type of SNPs, it's looking for mutational hotspots, looking for indels, insertions or deletions in the genome, maybe recombination sites where whole genes or gene families have moved in and out of an organism. All of these become really important for us to understand relationships and understand what's happened to these organisms to make them potentially virulent or resistant or just different somehow. Okay, so before we get too much further into sequencing, let's just discuss where sequencing is moving into the working laboratory. That could be the public health or the clinical or the environmental laboratory. So in the pre-next-gen world, and when I say next-gen, I'm going to say that a lot, that's next-gen sequencing. In the pre-next-gen world, a sample would come into the lab of which the goal was then what's in that sample, and so you try to isolate, culture out any organisms that are in that sample, and then once you get that isolate, then you try to speciate it using morphology or biochemical tests and even some molecular tests. Well, some of those molecular tests, like PCR, have become so useful that they can now be applied directly onto the sample. Now, these PCR tests can give us a little bit of information about an organism. They're very specific and sensitive, but we don't get a lot of information. We can say presence or absence of an organism or of a gene, but really we can only know very little about what's going on in a sample with just these types of molecular tests. What we need to do to find out more then is to take that isolate and do more analysis directly on the isolate. Hoping this will work. So we can, one of those analyses that we do is phenotype analysis, most commonly antibiotic resistance. There's other types of analyses, and what we end up doing, and I'll just stick with this. What we end up doing is a number of tests that can give us susceptibility or sensitivity to drugs, and that's going to give a lot of information about the organism itself, how can it be treated, what's moving around in the environment. We can also then do genotype analysis on the isolate using primers or enzymes, essentially to get some type of a profile, a molecular profile, or what we call a DNA fingerprint of an organism. And then we can infer from genetic similarity to genetic relatedness, and this is really how we have been tracking disease outbreaks for the last 20, 30 years. But now let's move into the next gen era. So we'll start with the isolate, and one of the things that we know that we can do is whole genome sequencing, and I'll talk a little bit about how we do this. But with whole genome sequencing, we get all of this data that we can essentially compile, put together, and now we can start to better understand what these organisms are, what's the content of these organisms, as well as then develop, I don't know if you guys can see, dendrograms or phylogenetic trees about true relationship between organisms based off of their full genomic content. But what's really exciting is actually now applying this directly to samples, not having to isolate the organism, but look at samples directly. And one of the areas that I see is a huge game changer, especially in the clinical world, is Amplicon sequencing. We heard a little bit about this today and probably elsewhere in this conference about Amplicon sequencing, where you actually just take the Amplicons that you amplify up from primers, just like PCR, but then sequence those to look at the genomic content within that. So you can look at a whole number of targets, a very large number of targets, dozens to hundreds, all at the same time within a sample, and look to see if that organism's present, and then look for important pieces of information in that organism along the way. And in this way, we can do very highly multiplex. And again, I'll talk a little bit more about that in a minute. We can also, one of the things that was talked about today earlier was microbiome analysis. And this type of Amplicon sequencing allows us to do a certain type of microbiome analysis where we can look at conserved genes across all bacteria or all fungi, sequence or amplify those up and sequence them, and that sequence data starts to tell us a lot of information about what's present in a sample. So then we can start to develop kind of microbiome profiles, abundance of the various organisms that may be present in a sample. And then the coup de grace would be whole sample sequencing, or you've heard metagenome sequencing, sequencing up everything in a sample, where we get all of the organisms that are present, plus if it's a clinical sample, and potentially even pre-produced food samples, we'll get the host DNA as well, whether that's vegetable or animal. We can sequence all that up and start to decipher and pull that apart. And this is very new. There's some things that we are very successful with, and there's other areas that we can only dream about where we're going to be in the next few years. When I say next-gen sequencing, or next-generation sequencing, we've been using that term now for several years, and it really applied from the movement from Sanger sequencing into essentially massively parallel sequencing, where we can get a lot of information. So with Sanger sequencing, we were getting kilobases, tens to hundreds of kilobases per run, bumping all the way up several orders of magnitude, doing up to gigabases per run. Which means that there was a lot of data starting to come out of these instruments. So it would take a fair amount of time to sequence, but then we would have so much data we'd have to really use high capacity or supercomputers to start to look at this data. So that's not very accessible to your average laboratory. But this has really changed now with where sequencing is going. We have tabletop sequencers which can run in a matter of hours and maybe a couple days. But then the data, and the data is still in the gigabase range, but we can do really targeted analysis. We can do analysis straight on desktop or laptop computers. We can use programs in the cloud. We can also still do the very large analysis of all the data, but we've become more targeted, more accessible, and hopefully more automated. I'll discuss that in a second. Another reason why sequencing is becoming more accessible is cost. It used to be off the charts expensive to sequence only 5-6 years ago, but now it's people have seen the comparison between Moore's law and what they call Carlson's curve, or just the cost of sequencing was dropping so precipitously it looked like sequencing was really going to be free. But we really see over the last couple of years that it's really leveled out. So sequencing has actually become very cheap and very accessible to do when you have a laboratory that's ready to sequence, but the cost has remained the same essentially over the past couple of years. Now, just to talk a little bit about the sequencing process, I'm going to only take a couple steps, and this is only for a couple of the different platforms that are out there. There's lots of ways to do this, and I'm not going to do, this is not a 101 on how to do sequencing, but just to think about the processes in the laboratory. First, there's the laboratory, or the library prep, which is essentially you just have to get your genomic DNA ready to be sequenced. One step of that is to essentially fragment or break up your DNA into small sequenceable chunks. And again, depending on the platform and depending on what you're trying to do, that could be anywhere from 50 base pairs to several hundred base pairs, or potentially even longer. And there's different ways to do that enzymatically or using sonication or other ways to disrupt the genome into the right size pieces. And then you add adapters onto the ends of that so that it can actually work in the sequencing system. In this case, I'll show attaching to a flow cell that allows then for some amplification and then further sequencing. The adapters also allow us to add indexes or barcodes onto our samples, so we can put multiple samples in together and then later pull them apart based off of how they've been barcoded. Now we have a library that's ready to move into the sequencing system. In one particular case, we'll talk about using Illumina here, but I don't work for Illumina. We just use a lot of their products, as do most people in the world of sequencing. Those prepped library pieces of genome now can be attached to the flow cell. They have the adapters on there. And then on the flow cell, essentially we're just going to amplify up clusters. So we have a lot of material now to sequence. And then once that's amplified, go through the sequencing process. And then essentially, as you can envision, reagents with adding in your nucleotides that are labeled, and if they hit the right spot and it's imaged, if it's not, then you won't get an image of it with the fluorescence. And then they just do this for as many cycles as you're going to sequence. So if you go 60 cycles of this, you're going to get 60 base pairs of sequence. And so that can take, about 60 base pairs will take about 3 or 4 hours, I should say about 4 to 6 hours. But then typically you're going to go 150, 300 base pairs. So this could go overnight or a couple days worth of sequencing. But within a couple of days, you're going to have a lot of sequence material, of which then you have a lot of data. So what now you've done is you've taken the sequence and digitized it, and really now have the digital version of the genome, of which you have to use bioinformatic tools to start to pull that all together. And this is just showing an alignment of several reads, and we have tools then that start to fit these jigsaw puzzle pieces together. As I mentioned, we can multiplex by adding in indexes into the adapters at the beginning of this doing the library prep, and that allows us, depending on what we're doing, to do dozens to hundreds of samples running them together in the same sequencing. So this really lowers the cost per sample. I'll discuss that a little bit more in a minute. But as I mentioned, this bioinformatic analysis can get quite complex, and I'm not going to go through all of what's on the screen here, but suffice it to say, part of what we need to do bioinformatically is deal with the data. How do we get it usable and ready? And that's making either aligning our reads to a reference or actually making assemblies of all of our reads. Once we do that, then we can really dive in and start to do the analysis. We can do phylogenetic analysis, and we can do gene content, looking for the causes of this having a new phenotype. We can do many different types of comparative analyses. There's a whole lot we can do with all of this data, and that just is only limited by the bioinformatic tools that are developed. And I mentioned, and this is actually an old slide, but there's a number of bioinformatic tools that are out there. In fact, we've developed many of these at TGen, as probably some others in the room have helped develop some of these tools as well that are very widely used. And some of them go away. Some of these are no longer in use, and there's probably about 2 or 3 times the list that's up here that we're actually using today. But our goal is to actually make these more and more accessible. So rather than have tools of which then you have to punch in a command line to make it operate, we just want tools that actually are just going to spit out a result once you put a sample into the sequencer. So that's really the goal of the bioinformatic community now, or one of the big goals, is just to really make this very accessible to the user. You don't even have to worry about the concept of bioinformatics, just plug and play. Okay, so a couple examples I want to run through as to how we're starting to use genome sequencing in the variety of ways that I mentioned. Certainly in public health, whole genome analysis for molecular epidemiology has become all the rage, and it's actually critical, and you're going to hear a lot more about how it's being used. But just a quick concept, when thinking about genotyping, pulse field has been used now for quite some time. It was one of the great inventions of the 20th century, and I hope you realize I said the 20th century. Now that we're in the next century, we really do need the next generation of tools, of which then are being developed at CDC and FDA and other places, largely based off a whole genome analysis. One of the reasons why is that where we can get good information and good comparative analysis between whole genome and pulse field to a certain degree, and in fact, if we look at Klebsiella, we can see that both will identify, say, this group that we call ST258. It's highly drug resistant, carries an important carbapenemase gene, and it's been transferred around the world pretty quickly. The problem is, if we look at this a little closer, pulse field versus whole genome, the whole genome we know is based off of the evolutionary model, so we are much closer to truth on relatedness of these organisms. And if that's closer to truth, how does that compare to pulse field? Well, we can see it's all over the place. The pulse field is actually giving us really bad information about direct relatedness between strains within this group. So we know that we're getting a lot more resolution than we're able to with pulse field, but that's understood. If you compare it to something else like MLVA, the multi-locus VNTR analysis was a recent tool over the last 10, 15 years that shows very good resolution and comparatively actually plays really strong against whole genome analysis. This study that we did with CDC, we were able to show, looking at past outbreaks, that MLVA actually starts to lump a couple outbreaks together, and in some cases splits out samples that actually truly are part of an outbreak into different groups. So we know that, again, we have much better performance with whole genome analysis, whole genome sequence analysis. And we can apply this now to different types of outbreaks here when we didn't have, say, an originating sample of a cluster of patients who got valley fever fungal infection from their transplants. We were able to show they had identical infections, therefore they had to have come from the same source. In a more recent case that we helped out with, we were actually able to look at what was thought to be an infecting source as well as all the patients. And when you compare Exerohilum, which caused the fungal meningitis in the injectable back steroid that caused many, many deaths and certainly hundreds, if not thousands of cases just a couple of years ago, we could show that the patients and the vials of which had the drug in it all had exactly the same strain of this fungus. So really important even for doing analysis back to manufactured products. Okay, as I mentioned, I think that Amplicon sequencing is a real game changer, and we've used it in a number of ways. One of those is to think about the importance of the escape pathogens. These organisms are highly multi-drug resistant, being spread around the world very quickly. They're really limiting our ability, the spread of these is limiting our ability to use antibiotics. And we know that, it's causing tens of thousands of deaths every year, and it's hospital spread, it's community spread. We're now seeing it actually in food organisms as well, or in food as well. So there's a number of ways that these are really being spread in the community, and it's critical to be able to track them and find them. One of the major problems, though, is that these things have a world of antibiotic resistance, hundreds of mechanisms of antibiotic resistance. It's not just looking for the MEK-A gene in staph. It's looking for literally hundreds of mechanisms. So how do we do that in a molecular environment? Well, we know that we can, with Amplicon sequencing, we can look at a large number of targets. And here we can look at literally hundreds of targets. We have working on a system where we're looking at right around 100 different markers that give us information, or 100 different assays that give us information on these markers. We can do this sequence analysis in less than 3 days. We're pushing it down to be able to do within 1 day, and down to be able to do tens of dollars per sample, hopefully closer to $10 per sample. So incredibly cheap, using better information than we've ever had available to us previously. And what we get is very high resolution information because we're just doing Amplicons. We get great coverage. This is just showing here at the top. This is looking at Klebsiella straight from a urine sample. So again, this is also, that's the other thing, is we're looking straight at the sample. So we can go from an environmental sample or a clinical sample or a food sample with this type of analysis. We have 10,000x coverage. So we know exactly what's in this sample for this target. And then looking at this, we know what species it is. We can look at SNPs within here to tell us if it's resistant. We can look at relationship between organisms as well. So we're not getting all of the whole genome, because sometimes that's just too much information for us. But if we look at targeted, we can do it quickly and we can do it cheaply. One of the other ways, I mentioned microbiome. There was great microbiome discussions this morning where you just target conserved regions within the genome that all bacteria or all fungi share, the 16S gene or the ITS gene, and then just look at the variability within that amplicon. Again, becomes very fast and very cheap. This is what we're seeing is really important in the human side, is understanding the microbiome that lives on us and in us in different body parts, and then hopefully being able to monitor changes. We're seeing that this is starting to be referred to as a new organ system of the human body and going to be critical to follow. But what about, and so for instance, we can look now at not only what are the different microbial components that occur within a patient sample, but what's the relative abundance of those, and then start to compare what's going on between patients. In this case, we're looking at cystic fibrosis patients, but what's much more exciting is looking at what's going on longitudinally. And so we can look at time points and then just watch to see what happened. Why is something causing a change in this overall profile? For us, we see this as moving towards the built environment and being able to monitor long-term what's going on, say in a food production facility or in a laboratory or elsewhere, and just being able to monitor that so that you know that changes in airflow or people moving in or products moving in may change the overall microbiome. And then the last is whole metagenomic sequencing. Again, this is looking at the entirety of the genetic material within a sample. You suck all the genetic material out using DNA extraction, and then test for whatever's there. And there's a whole lot of tools that need to come in to use this. This is very still cutting edge, not ready for routine use. But we're still finding that we can get a lot of information by looking at the entirety of it and then starting to parse it out. Again, here's one of these lung samples where we can look at, in the midst of all that genomic information, we can find the piling up of reads to give us the Staph aureus genome, so much so that we were getting 20 to 30x coverage of the entirety of the genome. And mind you, this is just one small fraction of all the DNA that we're looking at the time. And in fact, we can go in and look for the presence, and we see the presence of the MEK-A cassette within here. We know that this is a MRSA, which is a very serious problem with a lung infection, and therefore can provide rapid information among everything else that's going on within that sample. Lastly, mega-analysis. With this, we can do very large studies. I'm not going to talk about Genome Tracker at all because you're going to hear about that, but the FDA and with partnerships with CDC and state health departments looking at very large numbers of genomes. We're doing one where we're looking at large numbers of genomes in a very local place, looking at all of the E. coli in the community of a city like Flagstaff, so about 60,000 people. We're looking at all of the E. coli in human cases, plus all of the E. coli showing up in all of the food meat sampled multiple times, once every 2 weeks for an entire year. So what that's given us is 3,500 genomes just for one small space. So now we can really understand how these organisms move in a community. So the promise of NextGen for the applied laboratory, we can do detection and genotyping now. We can do antimicrobial resistance profiling and other phenotyping really easily now, straight from sample. Polymicrobial contamination and infection, understand the microbiome. Looking at host factors soon. We're starting to do that, but it's still way too early for us to make much sense of it. We can do 3 to 8 hour sequencing now, and soon 15-minute sequencing. The 4th generation instruments are out there. They're not perfect, they're not even great yet, but they will be, and this will soon be really almost bedside sequencing availability. And then, of course, what I think is what is necessary to really move this to the next level is having kit-based and automation, so it really is plug and play. Grab the sample, have the DNA extracted, it's run and prepped, run through a sequencer, and you automatically just get a report within maybe even just a few minutes, but likely a few hours. Final thoughts. NextGen is fundamentally transforming the way we look at pathogen genomics, and it should change the way that we look at public health and food protection. Advances in sequencing technology are decreasing costs and increasing the data. That abundance of data provides both a barrier as well as then a pathway for continued translation. And it's our goal as the scientists to find better ways to really make the best use of that data so that we can turn it into knowledge. And then this is just a stepping stone to omics, to a full omics approach for personalized medicine, environmental analysis. We're just talking about genomics, even metagenomics, barely metatranscriptomics, soon, pulling that together, the proteomics, the metabolomics, the methylomics. There's a whole field, and this is just really a stepping stone that's going to allow us to bring all of these pieces together. Acknowledgements, this ton of people have gone into the work of the data I've shown you up there. Our partners all over the world. I can't even mention them. I still wouldn't even give them appropriate credit if I did, because we have fantastic partners. And with that, thank you.